Hello everyone. So I welcome each and every one of you to this Unacademy NEET channel. And today we are going to go with the another session for the genetics. And I hope that I am clearly audible and visible to each and every one of you. Yes or no? Just let me know in the comment section. Am I audible? Am I visible? Great, great. So I am audible also and I am visible also. Now see. So today what I am going to do is that I came to know that we have missed a part which is non-allelic interaction, or it is also known as two gene interaction. Yes or no? So instead of doing the sex determination here, what we are going to do is that we are going to do that non-allelic interaction first. And when we complete that non-allelic interaction, then we are going to go with the sex determination. Clear to everyone? Yes or no? Let me know in the comment section if this thing is clear. Right? So we will do the sex determination also, but before completing the non-allelic interactions. Clear? Great. Great. So let's move on and let us talk about let's talk about the non-allelic interaction. You remember we discussed about the allelic interactions, yes or no? So what was allelic interactions? So in allelic interactions, we were seeing that if suppose this is a gene, okay, if suppose this is a chromosome, and in this chromosome we had, let's suppose, plant height, right? We had plant height, the gene for plant height, and second we have the another pair of this chromosome. Again in this also we had what? We had the same thing which is present here, which was the plant height. Yes sir. So in this what we were getting was, in this when these two alleles were interacting, right? Suppose this was a dominant allele, capital T, and this was the recessive allele, which is small t, and when these two were interacting, we were saying that they are genes interaction or allelic interaction. Yes or no? Which we have done in codominance and all those things. Yes or no? Did I said wrong something? Yeah. So which we have done earlier. Yes or no? Clear sir. So today what we are going to see is that, let's suppose this was what? This was the allelic interaction, which we have done in previous classes. This was allelic interaction. Allelic interaction. Now today what we are going to do is that today we are going to do the non-allelic interaction. Now sir, what is non-allelic interaction? See, let's have an example with us. So suppose this is one chromosome. Let's suppose this is a chromosome number 1. Let's name this. This chromosome is chromosome number 1. Yes or no? Okay sir. Now what? Now we have another chromosome, and that chromosome is, let's suppose, chromosome number 10. Okay, this chromosome is chromosome number 10. Clear? So I have taken two chromosomes. One is the chromosome number 1 and the other is, let's suppose, the chromosome number 10. So now this is so very much clear that these two chromosomes are different. Obviously, now these two chromosomes are different. One is the chromosome number 1 and the other one is the chromosome number 2. Yes, and if suppose that there is a gene present in chromosome number 1 which is A. Here a gene is present on chromosome number 1 which is A, right? And this gene is interacting with another gene which may be present on any chromosome, let's suppose chromosome number B. So now what is happening? The different chromosomes, the different genes are interacting, yes or no? Now different genes are interacting. So we can say that this is the intergenic interaction or non-allelic interaction. See, if it was not allelic interaction, so this A would have interacted with small a only. But now what's happening? This A is interacting with some other gene which is present somewhere else. It may be on second chromosome, it may be on the third chromosome, it may be on the first chromosome also. It may be on the first chromosome. Maybe this here on first chromosome there is another gene, let's suppose Z. And if A and Z is interacting, then also we will be calling it as what? We will be calling it as non-allelic interactions. Yes or no? So here alleles are not being interacted with each other. The genes, different types of genes are interacted. Now let us understand this thing again. Okay, let's suppose One step at a time, small step at a time, we are going to understand. Okay, if you are not able to understand this right away, I am damn sure that you will be able to understand within few minutes. Clear? Okay, now see. So let's take an example again. We have eye color. Okay, so eye color can be what? It can be blue. As per Honey Singh, it can be brown, right? Brown Monday and all those things. So eye color is what? Eye color we have various colors. Okay, so eye color basically is what? Eye color is being controlled by a gene. Yes or no? Now, the alleles for this gene may be containing brown color, right? Maybe containing blue color. Okay, and there is another character. Let's suppose your height. I'm just giving the example. It is not happening in practical life right now, that eye color and height is not showing the non-allelic interaction. Just to understand this, I am explaining you. So let's suppose our height. Okay, and if these two things are interacting with each other, let's suppose a person is having brown eyes and that person, brown eye person, is having tall height. So that means what? Here we are getting a confusion. The person who is brown is having tall height. That means these both are genes are what? These both genes are linked. So these are two different genes, but they're controlling each other, and this type of condition is known as what? This type of condition is known as non-allelic interactions. Now let me know in the comment section if this is clear or not. Any doubts anywhere, let me know in the comment section. Any doubts anywhere, let me know in the comment section. If there is no doubts, give me a thumbs up so that I can move forward. Yes, Shivesh, do you have any doubts? Today lots and lots of students are absent. Great. Should we move forward? Okay. So again, this thing was just for an example. In our human being, there is no relation between the eye color and the height. We have just taken this for an example. Clear to everyone? Okay, great. Now let's move on and let's talk about this two gene interaction in very much detail. Okay, great. So first of all, we are going to see that what is this two gene interaction. We saw that non-allelic interactions, here alleles are not involved, the different types of genes are involved. In non-allelic interaction, the very first thing which we are going to discuss will be what? It will be the complementary gene interactions. Okay, it will be complementary gene interactions. Okay, let me write here complementary gene interactions. Clear? Great. So now if I talk about this complementary gene interaction, what is going to happen here? See, complementary what? Complementary is, let's suppose that you are traveling in a metro all alone, so you will be very, kind of a quiet person. You will not talk with someone much. You will be behaving properly. But let's suppose you and your best friend is traveling in a metro, then so you will create a chaos. Yes or no? Yes or no? So in presence of best friend, in presence of some other genes, your character is being improvised. Your character is being affected. Right? Clear? So this is known as what? This is known as complementary gene interaction. So we will understand this again with a very simple example. See, this complementary gene interaction is being seen in sweet pea. Okay? Is being seen in sweet pea. Don't confuse this sweet pea with garden pea. Garden pea is different and this sweet pea is different. This is seen in sweet pea. Okay, everyone? Great, sir. Now see, if I'm talking about the sweet pea, what should I say? What is the scientific name? So the scientific name for this sweet pea is Lathyrus odoratus. Okay, Lathyrus odoratus. This is the scientific name for the sweet pea. Okay, now before going to the sweet pea, let us understand one more thing, and that thing is biryani. Okay, we'll make a biryani. How, sir? How we can make a biryani? See, To make a biryani, what do we need? We need something which is rice. We need something which is rice. Yes or no? Okay, sir. We need rice. Now, this rice has to be cooked first. Okay. Then then in this rice we will be adding some chicken. Yes or no? And if you are vegetarian, then add some veggies. Okay. So to make a biryani, what we are doing is that we are cooking rice first, and in that rice, what we are doing, we are adding some chicken or veggies according to your preference. And then we are again doing something, and then we are getting a biryani. Okay. Then we are getting biryani. Yes or no? Yes, sir. This much is clear. So can you see here, we are doing what? We are first making the rice. So when the rice is cooked, to cook rice, what do we need? We need heat. Yes or no? Let's suppose we need water, or we need a substance, or we need a process, which is, let's suppose, X process, or let's suppose C process. Okay. We need a process which is C process. What is the C process? Boiling of rice. Okay. And then to convert, when we added chicken, so we have to put dum. Okay. Like we put it in the vessel and we cook it in a low flame. That is called as dum. I don't know about much cooking, but yeah, this is what is done. So then again, some other process is being done. So this is what, let's suppose, P process. Okay, let's suppose P process is done. Now, if in case any one of the process is not done, we won't have biryani. Yes or no? If suppose you don't boil the rice, sir, I will make the biryani without boiling the rice. So biryani will not be made. I'll make biryani without putting the dum. So biryani will not be made. So that means to make biryani, you need what? You need both the process, C process and P process. Yes or no? Is this clear? Is this clear? Biryani, chicken biryani. Now, अब तुम्हें भूख लग जाए तो वो मेरी गारंटी नहीं है ना, खाकर आया करो मेरे क्लास में. Chalo. Now, let's move on. Let's compare this thing with the complementary gene interaction. So see, in sweet pea what happens? So there are two colors. In sweet pea there are two colors. What colors, sir? White color and the violet color. Okay. In sweet pea we have white color and VIOLET, violet color. How many colors do we have in sweet pea? The flowers of the sweet pea is having one white color, sir, and the other they are having a violet color. Okay. How they are prepared? Let's see here. How they are prepared, let us check here. So first of all, what happens? White is like rice. Okay. It's a raw product. It will happen. If nothing you are going to do, white is going to happen. Like if you are not going to make anything, white rice will be present, right? Uncooked rice will always be present. So treat white as a rice. If you do nothing, white to aayega hi aayega. White to will come and come only. Okay. Clear to everyone? Yes or no? Yes or no? Let me know in the comment section. Let me know in the comment section. Fast. Okay. Now see. So rice is there. Clear. So compare rice with the white color. Okay. Now to make violet color, there is some process to be happen. See. So to make white color into violet, there are two steps. First of all, there should be a C gene present. And this C gene will do something, and again here after the C gene interaction, after making the proteins for C gene will make the proteins, yes or no? Genes will make the proteins, yes or no? So after the C gene has made the proteins, the color of the flower is still white. Okay, the color of the flower is still white. Then comes the P gene. Okay, and this P gene is going to make what? This P gene is going to make protein. Yes or no? This P gene is going to make protein, and then this P gene, with the help of this P gene, what is going to happen? We are going to produce a violet color. Yes or no? So what's happening? To produce a violet color, you can take the screenshot. So to produce the violet color, sir, what things we are doing? we need C gene and P gene. If C gene is not present, the process will not take place. This process will not take place, and this will also not take place, right? If P gene is not present, so white color will not be able to convert into the violet color. Clear to everyone? Any doubts anywhere? Any doubts anywhere? Let me know in the comment section. If you have any doubts, let me know in the comment section. Okay, I hope that this thing is clear to everyone. Okay, great. Now one more thing. Here, in complementary gene interactions or non-allelic interactions, there is no change in genotype. Okay, in this, there is no change in genotype. I will write here. In this, non-allelic interactions, where is gone? Where should I write? Okay, I'll write here. In non-allelic interactions, no change in genotype. Okay, just remember this, genotype. The only changes which you are going to see will be what? It will be in phenotype. Okay, the only changes which you are going to see, it will be in phenotype. Clear to everyone? So phenotype will be affected. Phenotype will be affected here. Phenotype will be affected here. Clear to everyone? Yes or no? Yes or no? Great, great, Shivesh. Great. It's clear. Phenotype will be affected here. Okay. Now, sir, see. Now, what is going to happen? Just remember the dihybrid cross. Here, two gene interaction is taking place, right? Two gene interaction is taking place. So, remember dihybrid cross, we were studying the interaction like inheritance of two gene, like how these two genes get inherited to the from the parents to the offspring, right? And we made up unit, square and whole, and we got a ratio which was 9:3:1. Yes or no? We got this ratio, right? Yes? Anyone? Do you remember this? Do you remember this ratio? This was what? The ratio for dihybrid cross. This was the ratio for dihybrid cross. Dihybrid cross, normal ratio. Okay, normal ratio, nothing exception, nothing complementary, nothing, just the normal ratio. Okay, so normal ratio for the dihybrid cross was what? It was 9:3:3:1. Yes sir. Now in this 9, what we saw? In this 9 we saw that these 9s were what? These 9 were yellow and round. Yes or no? The 9 ones were yellow and round. Yellow and round. Yes or no? And to produce this yellow and round, what kind of genotypes you should need? There should be one dominant for yellow and there should be one dominant for capital R. Yes. So can you write here capital Y dash and capital R will produce what? Will produce the yellow round dominant thing. Yes or no? Yes. Yes, obviously. If suppose we have, I'll write this Y like this so that it won't get confused. Okay. So we have capital R and we have capital Y and capital R in this case. So at any cost, whatever genes comes here, if it comes capital Y, then also it is some small y, then also it is going to produce yellow and round. If here comes small r, then also it is going to produce yellow and round seeds. Yes or no? Because one allele is what? One allele is dominant, right? In a similar manner, if I talk about this 3, so in this 3, what is happening? We are getting yellow and wrinkled seeds. Yellow and wrinkled seeds. Yes or no? So, sir, yellow is dominant. Yes. So, that means capital Y will be present, right? But sir, here we saw that it is wrinkled and wrinkled was a recessive character. Yes or no? Wrinkled was a recessive character. So can I write here? It will be what? Smaller and smaller. Yes or no? it will be small r and small r. Then only na wrinkled is there. Then only wrinkled is there. Yes or no? Yes, is it clear? Then only the wrinkled is there, right? Yes, sir. Okay. Next we have what? We have again 3, which is what? Which is basically yellow, sorry, green and round. Which is green and round. Yes or no? Green and round. Clear? Okay, so for green and round, what should be the case? There should be small y, and there should be at least one capital R. Yes, and then we have 1. So here what's happening? We are getting green and wrinkled seeds. We are getting green and wrinkled seeds. Yes or no? We are getting green and wrinkled seeds. So if we are getting green and wrinkled seeds, it will be what? Small y, small r, small r. Yes or no? Small y, and small r, small r. Clear? Clear to everyone? So this is a general thing. This is a general thing. You should remember this. This is basically what? A general genotype for these kinds of phenotype. Yes or no? If you have 2 dominant, the phenotype will be dominant. If you have 1 dominant, so 1 character will have dominant phenotype, right? What did I say? 1 gene will have dominant phenotype. Yes. If you have again 1 dominant here, so the dominant allele of the gene which is having a dominant allele, so that character will be dominant. Yes or no? Okay, so green and wrinkled, no dominant alleles, so no dominant characters. Clear? Now this was normal. Now we are coming to the complementary gene interaction. Now let's see what is happening in the complementary gene interaction. Let's see what's happening in the complementary gene interaction. So let's suppose that the capital C gene is there, and this capital C is what? This capital C, so one second. Let us understand like this. So we have done the dihybrid cross. Okay, we have done the dihybrid cross for the 2 characters. So what ratios we are going to get here? We are going to get 9:3:3:1 ratio only, right? This is also dihybrid cross. Then we are going to get the same ratio, yes or no? Because there is no change in genotypes, right? So here in 9, what we are going to get? These 2 genes will be dominant, C and P. In 3, what we are going to get? We are going to get capital C and small p, small p. In this 3, what we are going to get? Like this, yes or no? And here though, capital C, and small p, yes or no? Okay. But sir, we saw that to make a violet color, both of this should be present. Yes or no? To make a violet color, both of them should be present. Yes, it's true. Both the dominant alleles should be present. Then only they are going to make the proteins, right? So see here. In this case, this is also dominant, this is also dominant. So the phenotype will be what? It will be violet. It will be violet. Yes or no? Clear to everyone? Great. Now here what's happening? This is small capital C and this is small p and small p. So see what we said, we discussed that to make a violet color, both the alleles should be what? Both the alleles should be dominant. So here one allele is dominant, so it will not make a violet color. The color will be white. The color will be white. Okay. Now we have this 3 also. So here also see small c is present, but capital P is present. But it's of no use. To make a violet color, we need both the dominant allele. So here also the color which we are getting is white. And ये तो कुछ भी नहीं कर पाएगा. This is to small c, small c, and small p. So this will be to white. ये तो white होगा ही होगा. Clear? Clear to everyone? Let me know in the comment section. Any doubts anywhere? Any doubts anywhere? Guys, no doubts? Everything is clear? Great. Great. Okay, chalo. If you don't have any doubts, let's move forward. Let's move forward. Now, So what will be the ratio here? What will be the ratio for violet and white flowers? Can anyone tell me in the comment section that what will be the ratio for this violet and white flowers? Anyone? What will be the ratio for this violet and white flowers? See, so for the violet, how many violet is there? 9 violets are there, rest all is white. Yes or no? So can you write here 9:7? देखो ना, देखो तो एक बार, 9 is violet and white is what? 3 plus 3, which is 6 plus 1, 7. So यहाँ पे the phenotypic ratio will be changed. So this ratio is for complementary gene interaction, which is found in Lathyrus odoratus sweet pea. Okay. So this is what? This is having 9. So this 9 is having what? This 9 is violet. And the 7 is what? The 7 is white in color. Right? The 7 is white in color. Clear to everyone? Any doubts anywhere? Let me know in the comment section. Any doubts anywhere, guys? No doubts? Great. If you have no doubts, then let's move forward. Let's move forward. So this was all about the complementary gene interaction. This was all about the complementary gene interaction. Now, let's move on and let us talk about the duplicate gene. Duplicate gene interaction. Okay, duplicate gene interaction. See, we are not going to study some of the things like detail mein we are not going to study like collaborative gene, inhibitory gene, polymeric gene interactions, but I'll make you write the phenotypic ratio so that if question comes, we can solve those parts. Okay, but the chances of questions coming from those are very less, but still we can't take the risk, right? Okay, so let's have the duplicate gene interaction now. What is this duplicate gene interaction, sir? See, if suppose that behalf of me, or suppose that I had a twin. Okay, let's suppose for an example, if I had a twin and instead of me, my twin brother is going to come here and taking the class. Will you be able to recognize? Maybe by face, maybe not. Okay, so what's happening here is that if suppose there is only one dominant allele is present, na, work will happen. Here what's happening in complementary was what happening? Both dominant allele should be present, right? But here in duplicate gene interaction is what? अगर एक भी होगा ना, एक भी dominant होगा ना, तो काम हो जाएगा. That means what? If suppose capital C किसकी तरह काम कर रहा है? कैपिटल P की तरह. So दोनों में से कोई एक भी होगा ना, तो काम हो जाएगा. This is duplicate gene interaction. Clear? Let's suppose that हम दोनों हैं. I have twins. Let's suppose I have twins and I and my twin brother is standing here. So we are doing the work. But if I go somewhere, he also can do the work. Okay? Clear? Clear to everyone? Any doubts anywhere? Let me know in the comment section. Okay, chalo. Let us understand this duplicate gene in very detail. So this duplicate gene thing is found where? It is found in the capsula. Okay? Example is what? Example is capsula. Shepherd purse, you know? Shepherd's purse. VIR87. Your name is also awesome. I liked your name. Okay, chalo. So we have this capsula, right? Okay, so we have this capsula. In capsula, we are going to find this duplicate gene interaction. So see, again remember, thank you so much, VR, right? Okay, so again let's remember this, the dihybrid cross, okay? Let's remember the dihybrid cross. So I'm not going to write again all the things, okay? I have explained it. So now I'm just going to write the ratios. So in dihybrid cross, we saw 9:3:3:1. Yes or no? This was coming the normal dihybrid cross, okay? So in normal dihybrid cross, we saw to produce this whole dominant character, we need what? We need capital Y. I'll write capital Y like this and capital R. Yes or no? Then for this, what we needed? We needed capital Y and small r, small r. For this, what we did? Small y and capital R. And एक number तो बिल्कुल ही, you are in class 9. Great. Great. If you're in class 9, it's great. You want to prepare for NEET from now only? Then it's awesome. Then it's really awesome. Okay, now see. So one is what? One is here. Like this is 1. Okay, this is 1, ratio 1. So in 1 we saw everything was recessive. So this will be small y, small y, smaller, smaller. Okay, clear to everyone? Both capital. Yes. Great. Great. Small y, small y, smaller, smaller. Okay, this was the normal condition. This was the normal condition. But sir, we are dealing with the duplicate gene interaction. We are dealing with the duplicate gene interaction, sir. So see, let's suppose there are 2 genes which are controlling the color, controlling the shape of the capsule flower. So see, here what's happening, 9, if I have 9 plants, so what will be there? It should be capital A, capital B, both dominant, so the shape will be triangular. Okay, both dominant, shape will be triangular. Now here let's have 3 ratios. So here what we are saying, that only one is dominant. Only one is dominant. Yes or no? Only one is dominant. If one is dominant here, what I said, duplicated, like this gene is what? This gene is a duplicate gene working as a duplicate, working as a twin for the other gene, right? For the other dominant gene. So इससे भी काम चल जाएगा. So यहाँ पे भी shape क्या हो जाएगा? It will be triangle. Just let's go back once again and let's check that complementary one. So in complementary one, what happened to my laptop? Huh? 1 second, guys. 1 minute रुक जाओ. Yes, this was the complementary. So see, here what was happening? एक dominant से काम नहीं चल रहा था. There has to be, to produce a violet color, 2 dominant must be there. But here what is happening? एक dominant है तो दूसरा dominant का भी काम कर जा रहा है. Clear? Now here also, again see here, this was small a, and let's have capital B, small b. Yes or no? So here also what will be? We will have a triangular shape of the flower. Why? Because one dominant allele is present. And इसमें तो कुछ भी नहीं होगा. Which one? Which one VIR? Which one I should say it again? And here small a, small b, small b. So the shape will be round. Yes or no? So here in capsula we are saying that if there is what? There is the presence of both the alleles. Sorry, if there is a single dominant allele, a single dominant allele is present, then also they are able to show the dominant character. But here in complementary gene interaction, what's happening? To show the violet color or to show the dominant violet color, both the genes should be dominant. Okay, both the genes should be dominant. So even one is controlling whole. Now, yes, this is correct. Even one dominant allele is able to produce all the phenotype. Yes or no? Sorry, the dominant phenotype. Clear? This was a case in duplicate gene interaction, but in complementary gene interaction this is not the case. And the people who thought that I am taking the class on sex determination, so guys, I am very, very sorry. I came to know that I have not taken the class on this 2 gene interaction. So instead of sex determination, I started this. I'll ask my team to update the thumbnail and the title. Okay, so don't worry. Okay, everyone? Yes, Akshay, Shivesh, clear? And VIR, is it clear? Great. Okay, chalo. Now next we अच्छा इसका ratio तो देखा ही नहीं हमने. Check the ratios. So ratio will be what? How many of them will be triangular in shape? So sir, 9 plus 3 will be 12 and 12 plus 3 will be 15. Yeah, I know this much maths, okay? Although I'm a biology teacher, but ऐसे +1 +2 तो करने आता है मुझे. तो 15, this one will be 15, yes or no? And this one will be 1, right? So 15 we have what? What is this cult? What is this cult? So 15 is what? 15 is triangle shape. 15 we are having, I'll write here, okay? By VIR, 15 is what? 15 is triangular in shape, and 1 is what? 1 is oval in shape, okay? So 15 is triangular triangular, and one is oval in shape. Yes or no? Yes or no? Clear to everyone? Great. So this was all about the complementary gene interactions and duplicate gene interaction, right? Now let's talk about the epistasis. This is very much important because if question is gonna come, it will come from here, right? This is a kind of complex thing. So whoever is seeing this video, stop doing everything and focus here. So we have epistasis. We have epistasis gene. Okay. Now what is this epistasis? See what's happening here. I'll explain you this phenomena. So this is of two types. One is dominant epistasis and the other one is recessive epistasis. Yes or no? The one is dominant epistasis and the other one is recessive epistasis. Okay. Dominant epistasis and the other one is recessive epistasis. Okay, so let us understand something about epistasis first. Okay, पहले epistasis समझ लेते हैं, फिर हम समझते हैं. अच्छा, resisting the function, what function we are here? पहले let us understand the term epistasis and then let's talk about the dominant and recessive thing. चलो, okay. Now see, a very good example I'll give you once again. What example should I give you? Okay, chal. Let's suppose that there are two groups. Okay, ऐसे schools में group होते हैं ना हमारे पास. Yeah. Yes. So we have two groups. Yes or no? What two groups? Like suppose that you have first benchers and the last benchers. Okay. First benchers and last benchers, and both are like, you know, they both are different organisms. First bencher are very different, last bencher are very different. Last bencher is like, whatever the teacher is teaching, I will eat my lunch, I will dance, I will cry, I will do everything in the class. And the first bencher is like, they are very studious. Yes or no? Yes or no? So now you have to decide which bencher you have to become, right? Personally, I was also last bencher, but I became a teacher. So rest is history. Now see, so in first bencher, let's suppose there are two best friends. Okay, there are two best friends, and among them, let's name it as A and B. And here what is happening? B is very powerful. Okay, B is very powerful. थोड़ा डोले शोले बना दो उसके. B is very powerful. Okay. In the last bencher also, what's happening? There are two person. Okay, there are again two person. Tuck 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 tuck, and let's name them as also A and B. Okay, A and B, or let me name it as small a small b. Okay, small a small b, right? So here what's happening? A is very strong. Let's suppose A is very strong. So fight is going to take place between this first bencher and the last bencher. दोनों लड़ेंगे एकदम, तलवारें निकलेंगी, है ना, खून बहेगा, दोनों लड़ेंगे. But wait, what happened is that there was a time where only like in first bencher both were present, A and B were present, but in the last bencher only B was present. Only B was present. So at this particular time, what's happening? At this particular time, this one is what? This one is dominant. So last bencher will lose the fight. Again, see what I said. I said that first bencher and last bencher is having a fight. Okay, so here what's happening? Case 1 or fight 1. In this, what happened? In first bencher both one was present, A and B were present, and in last bencher only B was present. So see, B was what? B was not strong. B was not strong. So when the fight is going to happen, B will lose. The last bencher will lose. In a similar manner, let's suppose that in case 2, what's happening? In case 2, they came to, you know, ऐसे बोलते ना, मैं तेरा भाई बदला लेगा. So यहाँ पे अब क्या हुआ? A अकेला था और कौन आया यहाँ पे? छोटा A आया last bencher से, right? So in this case, what is going to happen? First bencher will win the fight. So what's happening here, sir? We understood fight and all. What's happening here? See, so here one allele One gene is what? One gene is controlling the other character. It is suppressing the other one. The dominant allele is suppressing the recessive allele of different gene. See, again, let's have an example again, one step at a time, okay? I know the examples, toughest and important topics, please. Yes, I have planned it. Don't worry. See, so here what we are doing, that there is, let's suppose, I know that example would not made you a clear vision. After explaining this example, let's check out this example, then it will be clear for you, okay? See, so these are what? These are 2 chromosomes. Okay, sir, these are 2 chromosomes. Now what? In this chromosome, we have 2 genes, which is A and B, okay? The first bencher, okay? The first bencher, and let's suppose they are the last bencher, and they have the gene small a and small b, the last bencher, okay? Now what's happening is that this A1 is what? It is controlling this B. This first bench, the genes, that capital A and small a, the presence of dominant gene here is controlling what? Is controlling the recessive, the characters of the recessive gene. Yes or no? Yes or no? So who is controlling here? A is controlling, okay? A is controlling, so A will be called as epistatic gene. And who is being controlled here? B is being controlled here. So B will be known as what? Hypostatic gene. Yes or no? Let me know in the comment section if this is clear or not. So this A gene is what? This A gene is dominant over B gene. Like if A dominant is present, अगर एक भी dominant आ गया A का ना, तो ये B अपना character show नहीं करेगा, B डर जाएगा किससे? A से. Clear to everyone? बता दो भाई. Yes or no? Is it clear? Okay, let's move forward. And if you are liking this session, do share with your friends and families. No, why families, sir? Families will not learn bio from you, right? So you just have to share with your friends only. Chalo. Now let's move on and let's talk about the dominant epistasis and recessive epistasis. Okay. So first of all, let's have the dominant. Now things will be more clear. Dominant epistasis. See, if you will be like, na, ki I have to understand each and everything in one go, so that's not possible, right? You have to give time to certain topics. Step by step, you will able to understand everything. Now, let's have the dominant epistasis. So now if I'm talking about the dominant epistasis, so here you might be able to figure it out, like something dominant will be there and which will be, suppressing the recessive one. Okay, yes, that's true. This is the case only. And the example for dominant epistasis is what? Anyone in the comment section? Dominant epistasis. What's the example of dominant epistasis? So we are going to check the dominant epistasis in fruit color in summer squash. If you want the notes, if you want anything regarding anything, updates, when the class will happen, where it will happen, download the Unacademy app, and in that app search my name, HARSHKUMAR, follow it and join my community. Okay everyone? Chalo. Great. Now fruit color in summer squash. Okay, fruit color in summer squash. Now see what's happening. So see here, capital W, when capital W is present, or okay, let's understand like this. Again, we'll write 9:2, 3:3, 3:1. Okay, sir. Now here 2 genes are controlling the color, which is W and Y. Okay, which is capital W and capital Y, or small w or small y. Okay, so see what's happening here is that in 9 we have again both dominant. Yes or no? Both dominant, right? Okay, here we are having 1 dominant and 2 recessive. Here what we are having, here we are having both dominant, both recessive and one dominant, and here what we are having, both recessive and, okay. So sir, what's gonna happen here? What's gonna happen here? See, in normal condition, this is what, this is the dominant epistasis, okay. So if the dominant gene is present, if the dominant gene is present, one dominant gene is present, so it will what, it will produce its own phenotype. See, W is responsible, capital W is responsible for white color, and capital Y is responsible for yellow color. But since this is the case of dominant epistasis, ye capital W is already present here, so capital Y is not going to produce its phenotype. The flower color will be what? White only. Here also, see, here also what's happening? Here also one dominant is present, yes or no? Capital W is present, so here also the flower color will be white. But in this case, see, both the W are small, both the W are small, no dominant allele. So if no dominant allele is present, the capital Y will be like, अब मुझे मौका मिला है येलो कलर बनाने का, so ये हो गया yellow. And here also what's happening? Here also what's happening? W small, small w, small w, small y, small y. So this is going to give what? This is going to give you another color which is green, okay, which is green. Clear to everyone? Which is green, yes or no? See, why green, sir? See, small y to green deta hai na? Small y to already it gives green, yes or no? So here there is no dominant allele present here. So if there is no dominant allele present here, what is going to happen? The small y will produce its phenotype which is green. And here also what's happening? No dominant allele is present and capital Y is dominant, so it will produce its own color which is yellow. Yes or no? Yes or no? Clear? Is everything clear to everyone? Again, I am saying the ones who thought that I will be taking this examination class, I am very sorry. I thought of taking today only, but I remember that we have not completed this 2 gene interaction, so I am completing this. I'll ask my team to change the thumbnail and the title. Okay, everyone? Okay, chalo, great, very good. Now, let us move on. Let's move on. And how many of, let's discuss one more thing, which is recessive epistasis. Okay, let us discuss one more thing, which is recessive epistasis. Okay, recessive epistasis. Clear to everyone? Let's start with recessive epistasis. Okay, yes or no? Yes? Okay, chalo, great. So let's talk about the recessive epistasis now. Recessive means what, sir? The recessive allele is going to overcome or going to create disturbance in producing the phenotype of a dominant allele. Yes or no? Okay. So this is found in skin color or coat color of mice. Okay. Example is coat color, or I can see skin color in mice. Okay, in mice. Clear to everyone? Yes or no? Is it clear? Great. Great. If this is clear, then let's move forward. So now what's happening here is that, again, the genotypic ratio will be same, na? So let's write here 9:3:1. Sorry, the genotype will be same, right? So the genotype we will be getting will be what here? Capital A, capital C. Okay, let's have small a and small c. Then let's have capital A, small c. Then let's have capital A and small c. Okay, this is the case. This will be the genotype. And when both are dominant, so the color will be what? The color will be agouti. Okay, both are going to produce its thing. And this color is basically brown color, brownish color. Okay, brownish color is the agouti color, right? And here what's happening? This is what this is. This is recessive one, and this is the dominant one. Okay, this one is the recessive and this one is the dominant. So what is going to happen here? The thing which is going to happen in this case, we are talking about the recessive epistasis. So this gene will be affected in presence of this. So what will happen here? We will have the black color. Here will we have the black color, yes or no? And if I talk about the capital A and small c, in this case, what we are going to have albino color, that is white, which is white. Okay, here also see the recessive is present, and we will be going to have albino. What? Albino. So here in this case, the recessive epistasis is being shown. If the alleles, the recessive allele is affecting the phenotype of the dominant one, then it is known as recessive epistasis. Is it clear to everyone? Yes or no? This is what we have to learn in detail, but there are some other things which I am going to make you write. So one is what? One is the collaborative gene interaction. Collaborative gene interaction. Second is what? Second, we have inhibitory genes. Inhibitory gene interaction. Third is what? We have polymeric gene. See, this is what? This is, you can learn, but the chances of questions coming from this thing is very less. Okay, polymeric gene interaction, and then we have duplicate gene, which we have done. Okay, which we have done. So let's talk about this thing. So in this, what you remember is that ratio will be what? In collaborative, ratio will be 9:3:1. Okay, in inhibitory we will be having 13:1 ratio. Just remember the ratio. Okay, and 13:3, sorry 13:3. And in polymeric, what we are going to have? We are going to have 9:6:1. ये तो मुझे भी याद नहीं है, but I'll suggest you that you remember this. If in case question comes, then you can solve it. Okay, example क्या क्या है? Collaborative में comb shape in roster. Okay, comb shape in roster. Okay, then in inhibitory, what we have? Leaf color in rice. Leaf color in rice. Okay, leaf color in rice in inhibitory. Clear? And if I talk about the polymeric, so in polymeric we have example as fruit shape in summer squash. In summer squash. Okay, fruit shape in summer squash. Just learn this. Fruit shape in summer squash. This much is more than sufficient for you. So we have completed this thing, which is polymeric, what should I say? 2 gene interactions or non-allelic interactions. In the next session, we will be having the sex determination class, and after that sex-linked inheritance, and then the whole genetics, the Mendelian genetics will be clear, my dear children. Okay, so till then, bye bye everyone, and see you in the next class. I have a special class today at 10:15 on Unacademy app, where we are going to start with biomolecules. So if you are able to join, please join this special class. Okay, clear? Okay, chalo, bye bye everyone, see you in the next class. Do share this class if you liked, and comment for any kind of feedback, and if you have any doubts, then also you can comment. Okay, till then, bye bye everyone.